Great. Well, thank you very much for coming along. Good morning, good afternoon, good evening, wherever you may be, and welcome to this plenary session. This is the David Walton Antarctic Science Lecture, and it's my pleasure to chair this session and introduce our plenary speaker. First of all, I just wanted to briefly introduce myself and the Antarctic Science Journal and Bursary. I'm Kate Hendry. I'm a chemical oceanographer at the British Antarctic Survey, and I'm currently chair of Antarctic Science Limited. There we go. So Antarctic Science is a journal managed by Cambridge University Press, and currently under the steady hand of our editor-in-chief, Pete Convey, who's hopefully joining us remotely from Svalbard at the moment. As well as publishing on all aspects of Antarctic science in and around the Antarctic and sub-Antarctic, we use the profits from the journal to support early career researchers in the form of our annual international bursaries, but also educational grants and in sponsorship of APEX. If you want to find out more about the bursary and about the journal, then you can go to our website here or follow us on Twitter, and you can see our email address up there as well. So Antarctic Science was established back in 1989 by David Walton. You can see him here. David was an ecologist, spent a long and very distinguished career at the British Antarctic Survey, and also dedicated much of his work to making Antarctic science more accessible to the public. David sadly passed away in 2019, and in memory of his efforts in building the journal and in building the bursary scheme, the Antarctic Science Board decided to sponsor a speaker to attend each SCAR Open Science meeting. So we seek to invite speakers across a diverse range of backgrounds and subject areas, and those with excellent track records in Antarctic science and science communication. So with that, it's my pleasure to introduce our speaker for this year's SCAR Open Science Conference, Dr. Ceridwen Fraser. Dr. Fraser was awarded a Bachelor of Applied Science from the University of Canberra, followed by Bachelor of Marine Science from Macquarie University, before moving on to a PhD in zoology at the University of Otago, where she is currently an associate professor. She's got an astounding career record, including winning the ACT Scientist of the Year Award in 2016 and the Fennell Medal of the Australian Academy of Science in 2018. Her research combines molecular ecology with other subdisciplines of the geosciences to understand the processes that drive global patterns of biodiversity. And today she's going to talk to us about Southern Ocean diversity and connectivity, surprises in new directions. So I will hand over to you. Thank you very much.
Thanks very much, Kate. What a lovely introduction. I'm just going to start sharing screen. Wait. Launch. How's that looking?
That's looking great. Good,
great, thank you. So I've given this talk the title Southern Ocean Diversity, Surprises and New Directions. That's a very, very broad title, and there's a lot to cover under that umbrella. So I've only got half an hour, and I'll do my best at giving a bit of an overview and also a bit of a delve into the details of some of the work my group has been doing. I also wanted to acknowledge David Walton. What a great honour to be giving a talk in his memory. And I'm sorry I never actually got to meet him in person, but his career is quite clearly astounding and a great role model. And I wanted to say I'm very sorry that we're not attending this conference in person. SCAR is always a highlight. I love meeting all the other Antarctic researchers. And I was particularly looking forward to coming to India because I have a strong personal connection to India. My husband is Indian. We were married in India about 13 years ago or something now. And I have a couple of half Indian children who are giving a little bit of a headache at the moment because one of them's just got COVID. This is the first time COVID has reached our household, and the other is only 5 months old and hasn't let me sleep for more than 2 hours at a stretch for the last 5 months. So hopefully this will be a coherent talk despite my sleep deprivation. But yes, I'm sorry we're not in India, and still I'm excited that this conference is being run by India, and I'd love to hear from some Indian Antarctic researchers if there are opportunities for us to collaborate. I'm currently based at the University of Otago in New Zealand, and it's always beautiful and sunny like this. We're far down south. We're actually kind of almost sub-Antarctic. There are albatrosses and penguins in the city realm. It's not always sunny like this. This was us a few days ago after some torrential rain. That little creek running through the campus became a raging torrent. So of course the undergraduates got out their rubber tires and dinghies and went whitewater rafting down it, much to the horror of the powers that be. On to Antarctica. So Antarctica was, there are some discussions that Antarctica might have been discovered by people way back in the 7th century. There's oral history of Polynesians voyaging south and encountering frozen waters. So some say that this could indicate that Polynesians traveled as far south as Antarctica, or at least into the icy realm. There are some counter arguments that that's not very probable, because the quality of the vessels they would have been voyaging in wouldn't have withstood the Southern Ocean. Although I guess when you think of the Shackleton survivors voyage in a life raft back to South Georgia, maybe it is possible. It wouldn't have been very pleasant though. But really, the discovery of life in Antarctica kicked off in the 1900s in a big way, with that heroic age of Antarctic exploration, when a lot of the Northern Hemisphere nations turned their eyes south and looked at Antarctica as one of the last frontiers for discovery. And there were a lot of expeditions in the early and mid 1900s to try to discover more about this frozen land. Generally, when we think of diversity in Antarctica, we think of it as quite depauperate. When we look at pictures of Antarctica, there's pictures of ice, almost barren looking land. And of course, we know about this hypothesis of the latitudinal diversity gradient, where we think of high diversity around the equator, trending to lower diversity towards the poles. But as we are discovering more about the Southern Hemisphere, we're realizing that actually that latitudinal diversity gradient, at least for marine taxa, doesn't really hold for the Southern Hemisphere the way it does for the Northern Hemisphere. So this is from a paper that Stephen Chown led in Nature a few years ago. And we noted that actually Antarctic marine ecosystems are highly diverse. And the more we find out about them, the more we are astonished at their diversity. So certainly there's a history of every time there's a big expedition, we find a whole lot more about Southern Ocean biodiversity. Here's a graph from a paper by Hugh Griffiths et al. Back in 2010, just noting that every time there was a big expedition, there was a massive jump in the number of species described for the Southern Ocean. In 2011, De Breuer and Danis published a paper looking at the number of described species based on the Register of Antarctic Marine Species. And this is a really good resource you can go to online. It's kept up to date, so you can see how many marine species are known from Antarctica today. And while De Breuer and Danis in 2011 recorded about 7,000 or so known species, we're now in July, a few days ago, we're at a lovely 11,111 species, which is a very nice number. So it's going up all the time. And genetic studies are really improving our knowledge a lot, because every time we do a genetic study, we realize that sometimes what we think was one species is actually potentially more than one species, particularly for those species that have really broad distributions. And we thought they were circumpolar or circumantarctic. And then once we start doing genetic studies, we realize that actually there's a lot of diversity, biogeographic boundaries, and some of these could qualify as separate species. So for example, for the isopod Ceratosaurolus trilobitis, Held et al. In 2003 noted that there were probably at least 2 species within what we thought was one. In 2007, Nerida Wilson's group recorded at least 5 species level clades within the crinoid Pomacocrinus kurgalensis. And similarly for Orcamanella amphipods, Havermans in 2011 found around 3 or more species with distinct genetic clades. So we do genetic work and we discover more diversity. So if we look at what De Breuer and Danis described back in 2011, or that recorded the number of species around 7,000, in 2004, Gutt estimated that based on morphology, we probably have more than double that. And then if we consider that within each species, genetic studies are revealing more and more cryptic diversity, perhaps 2 or more species, then we'd be looking at double that double or even more. So there's a huge amount of undiscovered diversity yet to be recorded from the Southern Ocean. And of course, meanwhile, Antarctic marine ecosystems are under threat from changing environments and new arrivals. Antarctica's geographically quite isolated, and the Southern Ocean sweeps around Antarctica with the strongest current in the world, the Antarctic circumpolar current, heading eastwards. And there are these oceanic fronts, such as the Antarctic polar front, the sub-Antarctic front, the subtropical front, that encircle Antarctica. And we've always considered those as barriers to dispersal. The frontal zones are where colder water meets warmer water, and they tend to be subduction zones for water. So something drifting passively in the ocean, in theory, shouldn't be able to cross that zone. It should be pushed eastward with the direction of the current rather than heading southward. And back in 2012, I led a paper in Trends in Ecology and Evolution that basically said Antarctica is isolated. Nothing can get in or out except those really big animals that can swim or fly effectively, such as whales and flying seabirds. So I think I said, although a few highly dispersive marine species have been able to recolonize post-glacially, most surviving high-latitude taxa in the southern hemisphere appear to have persisted throughout glacial maxima in local refugia. And so I was comparing this to the northern hemisphere, where we see from thousands of studies, we see a very clear pattern of movement back towards the poles after the recession of ice, after the last glacial maximum. As land and ocean became available, species headed north to where the climate was more amenable. But in the southern hemisphere, we don't see so much evidence of that. And it could be because the Southern Ocean acts as a barrier to movement, and certainly it would act as a barrier to movement to a great many species. Genetically also, we see strong evidence of divergence between Antarctic species, for example, and South American species. This paper by Pullen et al. Showed that many marine taxa, marine invertebrates, are genetically very distinct in Antarctica from their sister lineages in South America, again indicating a long period of isolation. So we think of Antarctica as isolated, but we know, and there's an increasing interest in how species could be getting there. And invasive species are a really hot topic at the moment. How are people bringing plants and animals to Antarctica? So here's a series of examples of papers that have come out recently looking at the risk of invasive species, and in some cases, examples of invasive species reaching Antarctica. But there aren't too many marine species, or none really, that we know of yet, that have established in Antarctica that have come from elsewhere. Dana Bergstrom wrote a paper in Tree just this year that looks at the sorts of vectors that species could be using to get to Antarctica. So in this chart from her paper, I love this projection with Antarctica in the middle, and you see it really is surrounded by ocean and quite well connected to the rest of the world in that sense. Those red lines are shipping routes. So the shipping routes really connect Antarctica to much of the rest of the world. And that is a clear way that species could be getting in. Stephen Chown, Jasmine Lee in 2007 wrote a paper looking at one of the Antarctic vessels, the S.A. Agulhas, and they found a great many invasive mussels within the sea chest of the ship. So there is a very clear potential for species to get into Antarctica through ships that haven't been properly decontaminated before they get into Antarctic waters. There is a rule that ships need to release their ballast water well outside of Antarctic waters, and some recent papers have looked at how far away you need to release your ballast water to minimize the risk of any larvae or other propagules that are in that water getting to Antarctica. And of course, in terrestrial regions, we see that people often bring seeds of plants to Antarctica. Surprisingly, it's not the tourists that are the main culprits here, but actually scientists and research-based support teams that often carry seeds to Antarctica. So it is, Antarctica is under threat of new invasive species. But there's also potential for species to get to Antarctica on their own. And so here's a review paper written by a PhD student who worked with Stephen Chown and me a few years ago, Katie Moon. And she looked at connectivity around the sub-Antarctic and reviewed papers that assessed patterns of connectivity and found that really across lots of different species, there is strong evidence of connectivity among sub-Antarctic islands. That's through genetic work and other analyses. So these sub-Antarctic ecosystems, at least, are quite highly connected, much more so than we might have originally thought, given that they're separated by tens of thousands of kilometres of open ocean in some cases. And so now I'm going to talk a little bit about one of my pet organisms, Davilia antarctica, or bull kelp, southern bull kelp. In New Zealand, it's known as Rimu rapa. in Maori. And southern bull kelp is very, very buoyant. It's very large. It can grow up to 12 meters long and very robust. This is on the left here, you see a cross section through one of the fronds of De Villiers Antarctica. Sorry, I'll go back to that slide. And it's puffed up with gas, those cells, sort of gas-filled vesicles that make it highly buoyant. Circled in yellow here is the distribution of De Villiers Antarctica, mostly around the sub-Antarctic, New Zealand, and along the coast of Chile. And those populations are connected by the Antarctic circumpolar current. So for the last decade or more, I've been looking at how the Antarctic circumpolar current might connect those populations for De Villiers Antarctica and associated organisms. There's estimated to be about 70 million individuals of bull kelp, this southern bull kelp, drifting at sea at any time. And about 20 million of those are thought to still have their holdfast attached. And that's important because the holdfast, which is the disc that connects the kelp to the rock platform, is absolutely teeming with life. Here's a student holding up a detached holdfast that we chopped off the coast in New Zealand, and you can see many, many organisms living within there. There are crustaceans, amphipods and isopods, crabs. There are mollusks such as mussels, chitons, limpets, bivalves. There are polychaete worms. There are bryozoas. There are other algae. There are all sorts of taxa living inside these holdfasts. And so bull kelp can potentially act as a taxi service carrying species around the Southern Ocean. So with that in mind, I was quite astonished and excited when a colleague, Erasmo Macaya from Chile, sent me this photo in 2017. This is a piece of bull kelp lying on a beach, but it was taken in King George Island in Antarctica in January 2017. And Erasmo and his colleague Nelson Valdivia found two pieces, one on one beach and one on another beach on King George Island that year. That's astonishing because what we know of the oceanography of the Southern Ocean suggests that bull kelp should not be able to travel south to reach Antarctica. It should be swept eastward in the path of the Antarctic circumpolar current. So how did it end up on King George Island? We can use genetics to look at this. Now, if I'd been restricted to the sorts of methods that were available back when I did my PhD, this is a picture from the analyses I did during my PhD, we wouldn't have been able to answer that question because all around the sub-Antarctic, there is almost no genetic diversity in populations of De Villiers Antarctica, probably because these populations recolonized those islands after the last ice age. You see much more genetic diversity around New Zealand and Chile, but around the sub-Antarctic, there's almost no genetic diversity at all. Here, each color represents a different haplotype of cytochrome oxidase 1. But genetics has come a long way, and we now have really powerful genomic tools that let us to take a closer look at patterns in population and population connectivity. So here, using around 16,000 SNPs from throughout the genome, using genotyping by sequencing approach, I reanalyzed those same samples that I'd collected during my PhD and included the two samples from King George Island. And now that red haplotype from the previous slide breaks down into quite nice, neat geographic regions with those 16,000 SNPs. And using those data, we can see that one piece came from Kerguelen Island and one piece washed up on King George Island came from South Georgia. So that's really cool. We can now use genomics to pinpoint where dispersing or invasive organisms have come from. Even better, the following year, because people were now looking for it, we got another 25, 27 or so samples. Not all of them worked in sequencing from King George Island again. So a whole lot more kelp was found washed up on the beaches once people started walking around and looking for it. And we analyzed 23 of those, so making a total of 25, including the two from the previous analyses. And this time we could do much more powerful analyses because we'd sequenced a reference genome of De Villiers, and that allowed us to get more than 10-fold more SNPs out of the data. So we had a data set now of more than 220,000 SNPs and much higher resolution again to be able to pinpoint dispersing the sources of dispersing individuals. And so we see a bunch came from South Georgia, a bunch came from Macquarie Island, some from Marion Island, some from Kerguelen, and some from places that we can't quite identify. Now, we have most of the populations of De Villiers Antarctica in our reference system, but we don't have all. For example, we don't have any from Heard Island because nobody's been there for so long. So perhaps these rogue individuals have come from one of the unsampled populations. But still, we're getting really nice results using this approach, pinpointing sources of dispersing organisms. But like I said, de Villiers Antarctica shouldn't be able to get to Antarctica because of the Antarctic circumpolar current combined with Ekman transport, or the Coriolis effect. It should travel east and eventually head north. And all the oceanographic modelling we had done up to that point indicated that that should be the case. So I spoke with some oceanographers working at the Australian National University, Adele Morrison and Andy Hogg, and they thought maybe Stokes drift could be having an effect. Stokes drift isn't always included in models of drift for surface currents, because once you go down a metre or two, it's a negligible effect. It's the spiralling motion of a particle right at the surface, pushed by these wind-driven surface waves, and it really drops out very quickly once you start going down in the water column. And a lot of our models of ocean movement are based on drifters that are anchored to sit a couple of meters below the surface. So if something is sitting right at the surface, like de Villiers Antarctica, it's much more prone to be pushed by these wind-driven effects, the wind-driven surface waves and the wind itself. And when we include Stokes drift in oceanographic models, all of a sudden we get some particles able to cross those oceanographic barriers, those fronts around Antarctica, and hit the Antarctic coast. So they're shown here in blue and green, whereas all the ones that didn't reach Antarctica are shown in brown. So most of them head east and north as expected, but some are able to reach the Antarctic coast. So this is really exciting. It kind of overturns what we thought of Southern Ocean dispersal dynamics for things drifting right at the surface. When we do releases from South Georgia and Cape Horn, it's even more striking. In this case, many, many particles, still proportionally small, but a lot of the 4 million simulated released particles are able to cross those fronts and get to the Antarctic coast. So here we see particles, 4 million simulated particles released from South America, and quite a lot of them are able to hit the Antarctic coast. So this is strong propagule pressure. If we don't include Stokes drift, just as an aside, over here on the left, you can see what happens. None of the particles are able to reach Antarctica. So Stokes drift and the wind is clearly playing a very important role. But as we all know, Antarctica and the Southern Ocean are very, very windy. So we've made a little video about this. It's about 7 or 8 minutes long, and I'm not going to play the whole thing. I've just pulled out 1 minute of it, and hopefully this will work for you. I hope you can hear the sound. But this is a video aimed at communicating this sort of research to children, particularly children in New Zealand, and we've done a version in Te Reo Māori language, and Erasmo Makaya has also done a version with Spanish subtitles, which you can find on YouTube. But this is a little minute of the English version. If you want to see the full thing, just use that QR code up in the corner or type in the YouTube address. I think it's a lot of fun.
Up until recently, people thought that great cold continent was out of reach of rafters like me. It was like there was a big wall around Antarctica that we could never cross. The ocean currents swirling around the continent meant we'd never reach the icy shores. But when you're out here on the ocean, it gets really windy. Whoa! Sometimes my raft gets blown by the wind and we go in a different direction to the current. It's almost like surfing. Yeah, gonna hang 10, dude, righteous! So using this information, a team of researchers did some really clever computer modelling. They released 4 million virtual kelp rafts from each island in the Southern Ocean. The blue ones never reach Antarctica, but those yellow ones do. The researchers showed that the chances of a Rimurapa raft getting to Antarctica are tiny, way less than 1 in 100. But there are about 70 million Rimurapa rafts sailing the Southern Ocean on any day. So while the chances of one raft reaching the frozen continent are teeny tiny, because there are so many rafts in the ocean, hundreds could wash ashore in Antarctica every week.
So that was a fun video we've made. The one problem with it was they're quite upbeat about the idea of bull kelp and its associated animals reaching Antarctica. So I kept saying to them, this is not necessarily a good thing. Can we tone down how exciting it is? It's exciting research. but it obviously has huge implications for Antarctic ecosystems. But anyway, these rafting journeys that we've recorded are really long. If that kelp came from Kerguelen to King George Island, the shortest simulated trajectory that we ever achieved in the models was 20,000 kilometres, which is a phenomenal distance. The average of simulated trajectories from South Georgia was 25,000 kilometres. Some of them were shorter because some of them managed to get into the Weddell Gyre and come back up the back way. But most of them took the long way around, going around in the path of the Antarctic circumpolar current. So really long rafting journeys, but they're occurring in a biologically plausible time frame of a couple of years. So we now know that floating objects, including macroalgal rafts, can and probably often do reach Antarctica from northern sources. And so that means that with warming, there's a potential for these Antarctic intertidal ecosystems to drastically change from what they are at the moment, which they look barren, but they're not. You know, there's lots of life on those shores. It just tends to be life that shelters inside the rock crevices away from ice scour. But these could shift potentially into much more kelp-dominated ecosystems like we see in the sub-Antarctic in the future, if warming allows and if ice scour is reduced along some parts of the coast. And of course, if bull kelp floating at the surface can reach Antarctic shores, then many other things can too. There's a study from 2020 from Conchita Avila and her team looking at macrocystis kelp rafts found on King George, I think it was King George Island, maybe Livingstone as well, but same sort of part of maritime Antarctica. And those kelp rafts had holdfasts and which were teeming with invertebrate life. So we already see those passengers of kelp rafts reaching Antarctic coasts. And of course, plastics too can get to Antarctica this way. So inclusion of Stokes drift in oceanographic models can help to explain why we see so much plastic in Antarctic waters. This study from Kath Waller and colleagues shows, I think it's macroplastics in yellow and microplastics in green around the Antarctic coast and waters, and a study by Lacerda and colleagues use a bit of oceanographic modeling and also records of microplastics and macroplastics along the peninsula in the waters surrounding the peninsula to look at the possibility of Stokes drift and other processes pushing plastics into Antarctica. So this is clearly of great environmental concern. Also, bull kelp doesn't just carry invertebrates with it, but it can carry pathogens. So we've done some research in recent years looking at a protistan pathogen, Molinia, which affects bull kelp and other brown algae. It can jump from host to host, and it really creates these awful warts all over the kelp that it infects and can weaken the tissue. And some studies suggest it could be really detrimental to populations of kelp and other brown algae. So, you know, with pathogens like that reaching Antarctica, there is potential for them to host jump and affect native Antarctic species too. We now have some funding to look further at these questions. So we got a Marsden fund to look at a really interdisciplinary approach combining phylogenomics. So we'll do more genomic analyses, particularly for macrocystis, which we haven't analysed thoroughly yet, to resolve the sources of kelp rafts that are reaching Antarctica. If people come across kelp rafts, both macrocystis and Davilia, I would really love small fragments of the tissue so that we can include those in our genomic analyses. We're also going to do more modelling, both oceanographic modelling and environmental modelling, to look at how the ocean is helping kelp to get there, where along the coast the kelp might be hitting, and where there is potential for future establishment with climate change. And we're going to do physiological experiments. Some of these are already underway, manipulative experiments that can look at which taxa might be able to establish in Antarctica. So here's some of the team that are involved in this. We've got Miles Lamar, who works at Otago too, and Fabian Montiel and John Waters. They're all at Otago. Andy and Adele from the original oceanographic analyses. Kath and Hugh in the UK, and Erasmo, who's very involved in the phycological side of the research and has been getting a lot of the samples from Antarctica. Francis Perry is a master's student who's been doing these physiological experiments. Here's Parawaldeckia, a cute little amphipod that lives in kelp holdfasts, and she's testing whether they can survive at colder temperatures than they're currently used to. So a lot of physiological experiments are focusing on what happens with warming. Here we're looking at what happens when species are exposed to colder temperatures. Could they potentially survive in Antarctica if they've come from further north, from the sub-Antarctic, for example? Pluto is going to be starting a PhD very shortly on the macrocystis side of things, looking at the genomics and modelling. And Grant Duffy, sorry, that photo's become a bit stretched, sitting in his office here at Otago, has just joined our team, and he's doing a whole lot of exciting modelling, both in oceanography and particularly environmental and ecological modelling, to see where things might establish and where they might reach. So overall, I think there's lots of potential for exciting new research, but where we're moving in Southern Ocean research is really interdisciplinary and international research. We need samples from all over the place if we're going to get the big picture, so that involves a lot of international collaboration, and we want interdisciplinary studies that can combine different methods to really get into the processes that are driving patterns. So that includes genomics, as I've mentioned. Genomics is really powerful now. We can get really high resolution data without needing to know a lot about the genome of a species to try to pinpoint where dispersing organisms have come from, and also just broadly look at population connectivity patterns to understand barriers to dispersal that exist around the Antarctic. eDNA is also a really exciting emerging field that's starting to be used in Antarctic science. eDNA generally can only show presence or absence of taxa. And of course, in Antarctica, there are big questions around when you get a sign of presence of a species. Is it really there? Was it dead? Could it be hundreds of years old? Because Antarctica is a big freezer. But still, there are really exciting applications for eDNA to potentially track invasive or new species arriving in Antarctica, and to start to understand the ecosystems better when we might not have the time and resources to identify all taxa by eye, by morphology. Remote sensing is another key part of the future of science in Antarctica. There's been some nice work by Michelle Leroux and others looking at how remote sensing can be used to track particularly big organisms like penguins and seals. So we don't need to go out and count them individually with people on the ground, but can use remote sensing to count populations and see and monitor changes. There's a lot of increasing use of autonomous and remote controlled devices such as drones, ROVs. Here's Lee Tate from NIWA in New Zealand getting a ROV ready to operate in the Antarctic and take a look at benthic communities. And all of this can lead to integrative modelling that can help us to assess processes driving biological patterns. So we can combine data from different sources into hybrid models to start to pinpoint biogeographic patterns and processes around the Antarctic. We've just got a paper in press in Tree led by PhD student Gabby Kurdick and her supervisor John O'Tonkin, that's looking at how these different methods are leading to exciting new outcomes in Antarctica and how we can harness them for future research in Antarctica. But as I said, this is really, it's core that Antarctic research in future is transdisciplinary and involves a lot of international collaboration. We can't just work in our silos, our research field silos and our national silos. We need to talk to each other, share data, be very open about what we're doing so that we can get the most out of the research we're doing and work to better Antarctica's future. There are some really big programs that have launched. I know of a couple in Australia and New Zealand, so I'll talk about those, but there are probably others in other parts of the world too. One is Securing Antarctica's Environmental Future in Australia. This is a major tens of millions of dollars investment by the Australian government to bring together collaborators around Australia and some international partners to start looking at what's changing in Antarctica with warming. And similarly in New Zealand, the New Zealand government has funded a similar scale platform, the Antarctic Science Platform, bringing together Antarctic researchers across New Zealand and again some international partners. So both of these groups are doing some really exciting science and particularly focusing on changes in the future, what we can say about forecasting the future of both physical environments and particularly life in Antarctica. So that's really it from me, but essentially the take homes here are that we are harnessing powerful new technologies, and as we do so, it's revealing that Southern Ocean ecosystems are much more diverse and connected than we thought previously. But understanding and managing these ecosystems is going to require interdisciplinary and international research efforts. And I just want to make a note that as of this week, about 3 days ago, New Zealand's borders are finally open to the rest of the world. So I know for the past few years, whenever people have contacted me saying, can we connect, can we collaborate, could I come and work with you as a PhD student? The answer has always been, well, I don't know, because the borders are shut. We can't get out, we can't get in, and you can't get in. Now the borders are open. The borders are open to international students, and we can get out again. So there's opportunity for us to get out in person and start talking again and collaborating more widely. And with that, thank you very much for bearing with me, and I'd be happy to take questions. Back to you, Kate.
Thank you so much. That's absolutely fantastic talk. Really fascinating stuff, but also, as you said, really important as well, and really quite matters of concern as well. So something that we really need to be focusing on. And I really like the message, really agree with the message about interdisciplinarity and international collaboration too. So thank you, that's really great. Okay, so if you have a question, then please do put it in the Q&A, and I can read them out. I think I'll kick off with one burning question I have, which was, I don't think I knew that scientists were responsible for quite so much dispersal of seeds in Antarctica. That was quite horrifying. So if there's one or a small number of things that we could be doing better as scientists in Antarctica, what would you say those things would be?
Oh, I think, yeah, that's a good point. And it is shocking. I was also shocked at the results of that study. And I guess when I think of my own experiences working in the sub-Antarctic and Antarctica, I can see how it happens. We tend to think we're above reproach, that of course we're not going to be taking seeds or other tropicals to Antarctica because we're so careful. But are we really as careful as we should be? When you think about it, we're probably sort of pulling out that backpack that we took to Antarctica last time without really carefully checking all the pockets and every little corner of it. So I think those sorts of studies are really important for raising awareness that can then help us to improve our protocols, so that we really sterilize and clean all of our equipment before we go down there. That's very important. But I guess also, as globally, people are getting more and more interested in Antarctica. Nations are becoming more and more interested in research in Antarctica and setting up bases in Antarctica. More and more bases are being built at a quite astonishing rate. It's really important that we do everything we can when taking on those activities to minimize our impacts. The impact of research bases can be enormous. So just keeping that in mind, and everybody doing their very best to reduce activity, to reduce impacts. But I think some of these autonomous technologies are going to help with that in the future, where we might not need to be there in person quite so much or visit as frequently. We can set up devices that will track for us and gather scientific data without us necessarily needing to be there as often or to take so many people down there.
Brilliant. Yeah, and I guess that will also help with our carbon footprint as well. So another win there as well. Okay, great. We've got a couple of questions in the Q&A. Firstly, what is the most common invasive species found in Antarctica nowadays?
Yeah, thank you. So I can see both of these questions. They're kind of linked, aren't they? The other one is, have any invasive species ever been found away from coastal areas? I'm not the best person to answer this. I think I'd rather refer to somebody who's an expert on terrestrial invasive species, which I'm not. I know there are invasive species that have been found on land in Antarctica. Away from coastal areas, I'm not so sure. I mean, the trouble is people don't visit remote areas in Antarctica very much. So new research is showing that a lot of what we do in Antarctica is focused around research bases. So we tend to get a lot of data from close to research bases, not a lot of data from further away. So I'm not sure we would know if invasive species are establishing much further away. But as you move away from research bases, there isn't so much ice-free land, I think, to colonize. There are invasive species that have established in Antarctica. I believe there's a grass, but to be honest, I'm not sure of the name, so I'm not going to delve into it and try to bluff through that.
No worries. Thanks. Okay, so if you have any other questions, please put them in the Q&A. In the meantime, I'm just going to carry on because I have lots of questions. So if that's okay, I'll ask another one. So it's really
Rachel's
interesting.
Rachel's got an answer there.
Ah, great.
Sorry, carry on.
Great. So it's really interesting to see the results in the modelling about the Stokes drift that floating things can get across those oceanographic barriers. And I think we sort of think as scientists of those oceanographic barriers as being these completely impervious things, and especially even going back in geological history. So we think about the establishment of the circumpolar current around Antarctica, millions and millions of years ago, as when these when many organisms would have diverged. And so do you think your work actually has implications for how we actually interpret the fossil record as well, and how we can actually what we can say about the divergence based on the fossil record of these oceanographic gateways?
It's hard to say. I think it's estimates of when the Drake Passage really opened up and the Antarctic circumpolar current got really up and running range from 23 to 40 or so million years ago. So if we're going back further than that, Antarctica was still physically quite well connected to the rest of the world. We didn't have that very strong circumpolar current and the Southern Ocean blocking movement of species. But within those last 20 to 40 million years, the Southern Ocean probably has acted as a barrier to some taxa, but anything that can float at the surface, I would say, is going to be pretty well able to get across, and probably has been able to for a very long time. What's stopping them establishing at the moment, I imagine, is the extreme environment of Antarctica. It's extremely cold. There's a lot of ice scour. It's not somewhere that something rocking up from New Zealand, for example, is going to necessarily be able to establish. But that is, of course, changing with rapid warming. So part of what we're going to try to model is, will there actually be habitable land for some of these taxa in the future, in the near future or the distant future?
Great, thank you. Okay, we have a couple more questions. Have Antarctic scuba divers or ROV surveys shown any evidence of bull kelp becoming negatively buoyant and sinking as detritus?
So there's some nice research coming out of Chile, Martin Thiel's group, for example, that's looking at the longevity of rafts floating in the ocean. And Rothausler's papers show that over time, yes, we do get kelp deteriorating and sinking, but in the colder waters, it takes a lot longer. So as kelp heads up north, it's much more prone to breaking apart, deteriorating and sinking as detritus. But down in the south, it's quite happy and can potentially survive for years at the surface. It is nutrient limited though, out in the open ocean. So exactly how long it survives, we're not sure. But I think if we are seeing kelp washing up on Antarctic shores, we know it can get there. Modelling suggests it takes at least a year or two to get there. So that suggests it can survive a year or two pretty happily. The kelp that we have found on Antarctic shores, Erasmo has examined it and it still appears to be reproductively viable.
Great, thank you. And lastly, has there been an effort to introduce standardized guidelines to avoid the introduction of invasive species?
Yes and no. I mean, there's certainly guidelines around. As you know, with Antarctic science, different nations tend to manage their environmental permits and processes independently. So if I was, as a New Zealander traveling to Antarctica, to any part of Antarctica, I would apply through New Zealand, I believe, to get my environmental permit. So how well those protocols are adhered to by different nations really depends on the people who are both checking those and issuing those permits, but also who are the people who are traveling and how well they stick to them. There isn't a lot of policing of Antarctic activities, I suppose. So if you were to break the guidelines, break the rules, I'm not sure there are huge consequences at the moment. That's something, I suppose, that could be discussed.
Great, thank you. So I think unless there are any other questions, I think we should probably let you go and hope that the COVID situation is all right in your household and let you get on with things. But thank you so much again for a really fascinating talk. I learned a lot, and thank you very much to everyone who came along in the audience as well, and thank you for the questions.
Yes, thank you. Thank you for chairing, Kate. That was wonderful. And I'm sorry, I am a little bit brain dead today from sleep deprivation and stress. It's fine. I feel so
That's
silly
fine.
because the rest of the world has had COVID for ages, of course, but we only got it here in Dunedin in February. That's when it arrived in Dunedin. So for us, it's kind of, oh, the pandemic's arrived and now it's reached our house for the first time. Luckily, we're all heavily vaccinated.
Great. Well, thank you again, and
Thank you.
enjoy the rest of the conference.
Thanks. You too. Bye.
Take care, everyone. Bye.